Hello, my name is Rita Mormando, and my project focuses on determining the presence of JC polyomavirus in the urinary tracts of women with UTI-like symptoms. At this time, my team and I have no affiliations to disclose, and the funding for this project was done so as indicated here. So to start off, we'll be going over some general background about the subject. Current research has shown that there are microbes in the bladders of women with and without lower urinary tract symptoms. So in both healthy and unhealthy bladders, bacteria and microbe communities will in fact be found. We know that most acute UTIs are caused by an abundance of uropathogens, as seen in this figure below. They enter the bladder, and it's usually a common consequence of dysbiosis in the urinary tract. And they cause some form of distress when there's abundance of these organisms. There are some commonly known causes that contribute to various forms of UTI symptoms, but it's important to note the fallbacks that arise with UTI treatment specifically. About 30 to 44% of females with an initial UTI will in fact experience a second UTI, and with each UTI, the risk of another increases. So testing and treatment guidelines for chronic UTIs at this time are inadequate or simply do not exist in most parts of the world, which means that even when doctors want to help, they generally don't have the resources or guidelines they need to be able to. And in short, a significant number of females move through these escalating stages of diagnosis as antibiotic treatment fails to cure them and testing fails to find a cause. So it's clear from this that there is a real need to understand more about this occurrence so we can find better ways of treatment. We have known since the 1950s that there are viruses in the urine, and among those is polyomavirus JC. It's also known as JCPyV, or simply just JCV, so for the purposes of this presentation, I will be referring to it as JCV moving forward. We know from previous studies that JCV is a resident of the kidney in about 20 to 60% of older adults and is shed in the urine. But despite knowing about it for 70 years, we don't really know why it's there and what it's doing. So how do we tie it back into the UTI incidence? We have found that while conducting previous research that JCV is more frequently found in postmenopausal women with overactive bladder symptoms. And since there's some similarity in those symptoms with UTI symptoms, we found the goal for this study to determine the prevalence of JCV in the urinary tracts of women with UTI-like symptoms. And for this presentation, we broke this down into two steps, the first being through PCR analysis and the second through bioinformatic analysis. So for the PCR analysis, we took 53 urine samples from women with UTI-like symptoms and 30 urine samples from women without UTI-like symptoms, and these were both taken from a previously approved IRB study. The DNA was then extracted from the urine samples, and JCV was then detected by PCR using designed primers. From this, we found that of the 83 total samples for the PCR assay, 4 of them came back with a positive JCV mark. So roughly 7.5% of the population were able to detect JCV. And this was specifically only in women with UTI-like symptom cohort. So we found that there were no positive detection results for JCV in samples without UTI-like symptoms. And moving from this, we would like to include a higher population to extract DNA from in order to come to a more refined population occurrence and to then include the symptom data or metadata in the analysis to get a deeper understanding of the role JCV may have in accordance with these symptoms. So for the bioinformatic analysis, we took raw reads from the 2 studies listed here. And it's important to note that to date, there are only 2 shotgun metagenomic sequencing studies that have been conducted of the urinary viral community of individuals with UTI-like symptoms, and there are these 2 studies found here, so we figured it was the most imperative and important to include these in our study. We found the raw reads from these studies on the NCBI SRA database as well as the iMicrobe database, and we were able to take 54 uroviromes from individuals with UTI-like symptoms and 10 uroviromes from individuals without UTI-like symptoms, and we were able to map these to the JCV reference genome to determine the presence of a virus, and the reference genome accession number is listed here for your convenience. From this, we found that of the 64 total uroviromes, 9 had successfully mapped to JCV, so about 16% of the population. And again, this was specifically with individuals with UTI-like symptoms. None of the urovirome data sets from individuals without UTI-like symptoms detected any source of JCV. And we were able to determine the prevalence of JCV through a coverage graph that is shown here as an example of what it would look like. Coverage gives us a way to measure the sequencing identity. And as you can see, the entire genome is represented along the x-axis here with evenness throughout. So having coverage over every gene makes this a positive marker for JCV. And we were able to use this as a baseline for determining JCV positives. So moving from this, our study suggests that the incidence of JCV in the urine of women with UTI-like symptoms is in fact greater than in the urine of women without symptoms. And we were able to confirm this both using PCR and shotgun metagenomic sequencing of urinary samples. Moving forward, further PCR-based testing and urovirome sequencing of women without UTI-like symptoms is needed to really ascertain if JCV is shed in the urine as a biomarker of infection. But overall, this is a critical first step in ascertaining if JCV contributes to UTI-like symptoms and/or is a consequence of infection. So with that, thank you for listening to this presentation, and I hope you have a good rest of your day.